Hello everyone. Today we'll discuss about the digestive system. What is digestive system? What are the organs present in this? What are the functions of different organs? And what are the function of the digestion? And how it is going to be processed? Okay. So everything we're going to learn in this system. Okay. Right. And come to the system. This before going to the system, so what is digestion? We should know about this. Okay, so the digestion is nothing but the substances, the large substances which we are taking in by mouth, those are break down into the small particles is called as digestion. Okay, the large molecules or the large substances break down into the simple chemical components is called as digestion. This digestion process by the so many organs and including of the all those are organs, it make a system that is called as digestion here. Okay, right. And next come to the system and what are the functions of this system? If you see the functions of this system, so we have ingestion and breaking down and the transportation of the nutritive substances and what we are taking in, okay, transportation of that. And next one is secretion of the hormones, and by that there is a digestion process. And next one is digestion. So after the secretion of the hormones and the chemical compounds, okay, and the acids and everything, alkalines, and because of all this, how the digestion process is going to be done. Okay, so all this. And next, absorption. After completion of the digestion, so how that material, that chemical components are absorbed into the body. And next one is removal. And after the metabolic actions and after the process of digestion, the wastage, how it is removed from the body, all this are the functions of the digestion. Majorly, the functions are divided into 4 types, so that can be remembered as the idea. IDEA, in the sense I for ingestion and D for digestion, another E for egestion, and next one is absorption. These 4 are the major function of the digestion. And next one is, if you see this diagram, okay, so we'll come to know different organs of the digestive system, what are involved in this system. Okay, if you see that system, it starts from the mouth. Okay, so mouth and the teeth, next tongue, and next uvula, and after that pharynx, next larynx, and after that esophagus, next stomach, and next liver, and next pancreas, next small intestine, and the large intestine. All these are the organs involved in the process of the digestion. And next, if you see the functional anatomy of this digestive system, so what are the organs are there? There are 2 types of the organs are there. One is primary organs, which are directly involved in the process of the digestion, is called as primary digestive organs. In this primary digestive organs, we have, so the actually digestion takes place in that organs, that is called as primary digestive organs. So what are they? What are that organs in the sense? The first one is mouth, and next one is pharynx, and next one is esophagus, and after that stomach, small intestine, and the large intestine. All these are the primary digestive organs, in which organs the digestion process directly taking place. These are the primary organs. And next one is accessory digestive organs, which influences that digestive process. These are indirectly participated in the digestion process, like in the sense the food material or the food content not enters into that organs, but the secretion of this accessory glands or the accessory organs are enters into the primary organs, and the secretion enters into the primary organs, and there the digestion takes place. Okay, and what are the organs in the sense? One is the teeth. Okay, so by which masticate. Okay, so breaking down of that food material, and next tongue, which is helpful for the chewing, and next breaking down of this food material in the mouth, and next salivary glands, so which secretes the saliva, and next one is exocrine part of the pancreas which secretes the pancreatic juice, and next one is liver, so in which the bile secretion, and next one is gallbladder, and the concentrated bile is secreted from this, and the bile is stored in this gallbladder, and after that next one is the walls, so the walls of the GIT or the gastrointestinal tract. If you see these walls of the GIT, so there are 4 layers are present. The first one is mucus layer, second one is submucous layer, third one is muscular layer, and the fourth one is serous layer or the fibrous layer. Mucus, submucous, muscular, and the serous layer. 4 layers are present, inner to outer. 4 layers are present in this, okay, right. And if you see this diagram, we'll come to know so what are the layers are present and the arrangement of that layers. Okay, so first innermost layer is the mucus layer, and next upon that there is submucous layer, and upon that muscular layer, and next one is, and the uppermost or the outermost layer is the serous layer. Okay, and next one is the gastrointestinal tract is supplied by the nerve fibers. There are 2 types of the nerve fibers are supplied to the GIT, and one is intrinsic nerve supply and another one is extrinsic nerve supply. First come to the intrinsic nerve supply. If you see this intrinsic nerve supply in this, so the intrinsic nerve supply by the airbugs nerve plexus and another one is mucinous nerve plexus. Okay, airbugs nerve plexus and the mucinous nerve plexus, and extrinsic nerve supply by the sympathetic and the parasympathetic nerve fibers. Okay, right. If you see this intrinsic nerve fibers, okay, airbugs nerve plexus are present in between the submucous layer and the muscular layer, and the mucinous nerve plexus are present in between the muscular layer. That in the sense muscular layer is divided into 2 parts. One is longitudinal and another one is transverse muscular layers, circular muscular layer. Okay, so longitudinal and another one is circular muscular layer. In between these 2 layers the mucinous nerve plexus are present. Okay, right. And next is organs. Come to the primary organs. The first one is mouth. Okay, this mouth contains teeth and tongue, and next is uvula. If you see the diagram, we'll come to know what are the parts involved in this. Okay, teeth, and there are 32 teeth are present in the mouth, and 16 on upper jaw and 16 on lower jaw, so which are helpful in the breakdown of the food material. And next one is tongue, which is helpful in the taste of the food and at the same time, so during the mastication process. Okay, so the food material thrown into the in between to the teeth, upper jaw and the lower jaw. Okay, so that is what that. And next is oral cavity is present, and next uvula is present. Uvula is very useful so to move that food material inside of the tract, GIT tract. Okay, right. And next one is functions of the mouth. If you see the functions of the mouth, the ingestion, and next one is chewing of the food, and next one is appreciation of the taste, so the taste come to know, and next one is transfer of the food from the mouth to pharynx, that is called as bolus it is, and next one is speech, so what we are talking, okay, that is by the mouth, and next one is smiling and other expressions. Okay, after this functions of the mouth, next one is the salivary glands which are present in the mouth. The salivary glands are most important in the formation of the bolus, and which are easy to chewing and for the mastication process and the formation of the bolus. These salivary glands secretes, actually it secretes saliva. Okay, there are two types of the salivary glands are present. One is major salivary glands and another one is minor salivary glands. If you see this major salivary glands, we have three types of the major salivary glands are present. One is parotid gland, and next one is submaxillary or the submandibular gland, and third one is sublingual gland. Okay, three types of the glands are present. If you see this diagram, we'll come to know where these parotid glands are present and where the sublingual glands are present and where the submandibular or submaxillary glands are present. Okay, right. And if you see the how these secretions comes into the mouth, in the sense, if you see the parotid gland secretions are directly not enters into the mouth. The secretion of the from the parotid gland is enters through the Stenson's duct, and through that Stenson's duct that enters into the mouth. Okay, so parotid gland secretion into the Stenson's duct and then it comes into the mouth. Okay, and next one is submaxillary gland. The submaxillary gland secretion pours into the Wharton's duct, and that through that Wharton ducts that enters into the mouth. And next one is sublingual gland. The sublingual gland secretion is poured into the duct of Ravenous or the Bartholin glands, and through that duct of Ravenous, okay, the secretion poured into the mouth. Okay, and next one is, if you see the minor salivary glands, the minor salivary glands are situated in the lingual mucus gland, and next one is lingual serous glands, and next one is buccal glands, and next one is labial glands and the palatal glands. Okay, the major salivary glands are majorly in three types. One is serous glands, and next one is mucus glands, and next one is mixed glands. The serous glands and the mucus gland and the mixed glands, these are in three types. So based upon the type of secretion, these are divided into these three types. Okay, serous, mucus and the mixed glands. The serous glands are secreted from the serous cells, and the thin and watery type of saliva is produced from this type of the glands, and the parotid glands and the lingual glands are the serous glands. Okay, and next one is mucus glands. The mucus cells are present in this type of the glands, and the thick and viscous type of saliva is produced from this type of the glands, and the lingual mucus glands, and next one is buccal glands, palatal glands are the purely mucus glands. Okay, and next one is mixed gland. Both the serous and the mucus cells are present in this type of the gland, and the submandibular or the submaxillary glands and the sublingual glands are the, and the labial glands are the mixed glands. That in the sense both type of the cells are present, serous cells and at the same time mucus cells are present in this type of the glands. Those are called as mixed glands. Okay, and next, if you see the properties of saliva, the saliva volume is about 1000 to 1500 ml per day, or the 1 milliliter per minute. The secretion or the volume, okay, that is 1 liter to 1.5 liters per day. Okay, and next one is contributed by the each major salivary gland. If you see that the parotid gland contributed about 25% of the saliva, and submaxillary gland is about 75% of saliva, and next sublingual gland is about 5% of the saliva. So parotid 25%, submaxillary 70%, and the sublingual 5% of the saliva is produced from these major glands. Okay, and next one is reaction. So it is completely acidic reaction with the pH of 6.35 to 6.85 it is. Okay, and next the specific gravity of this is about 1.002 to 1.012. That is the specific gravity or the density of that saliva fluid. Okay, and next one is tonicity. It is completely hypotonic solution. It is hypotonic to the plasma. Okay, and next one is functions. If you see the functions of this saliva, it is very useful, and it is one of the digestive juice and the preparation of the food for the swallowing. And next one is appreciation of the taste is also one of the important function in the saliva. And next one is digestive functions. It digest the particles, will come to know. And cleansing and protective function of the teeth. Okay, and next, role in the speech. Okay, if there is no saliva in the sense, we can't talk actually. Okay, right. And next one is excretory function, is also one of the important function of the saliva. And next, regulation of the body temperature and the regulation of the body water balance. Okay, and the salivary amylase, which is secreted in the salivary glands. And the next one is, it converts the starch into the maltose. Okay, salivary amylase converts the starch into the maltose. And next one is, maltase is an enzyme which converts the maltose into the glucose. Okay, starch is converted into the maltose by the salivary amylase. Okay, and the maltase is an enzyme which converts the maltose into glucose. Okay, and next one is lingual lipase. This is an enzyme which converts the triglycerides of the milk fat into the acids and the diacetylglycerol. Okay, right, that is what that. And next, if you see the composition of the saliva, there is 99.5% of the water is present and 0.5% of the solids are present. And in these solids, we have organic substances and the inorganic substances are present. If you see the organic substances, there are enzymes and other organic substances. If you see the enzymes, amylase, maltase, lysozymes, and the lingual lipase, phosphates, carbonic anhydrase, and calcarein. Okay, and next one is other organic substances like mucin, albumin, protein-rich proteins, proline-rich proteins, and next lactoferrin, immunoglobulin A, and next blood group antigens, free amino acids, non-protein nitrogenous substances, and next urea, uric acid, creatinine, so xanthin and the hypoxanthin or the other organic components are present. And the inorganic components like sodium, potassium, calcium, phosphorus, chlorides, and minerals, anions and the cations are present in this. And next one is gases like oxygen, carbon dioxide, nitrogen. Okay, and normally the glucose is absent in case of the saliva, but it is found in case of the diabetes mellitus. Okay, and next is, if you see the regulation of the salivary glands and the salivary secretion, the facial nerve, hypoglossal nerve, and the glossopharyngeal nerve, three types of the nerve fibers are involved, and at the same time both sympathetic and parasympathetic ganglions and the preganglions and the postganglions are also regulated this salivary gland secretion. Okay, and next one is the applied aspects of the salivary glands. Xerostomia in the sense, completely stoppage of the salivary secretion is called as xerostomia. And next, drooling in the sense, continuous salivary secretion from the mouth, and that is in the sense there is no regulation, it is continuously the salivary secretion is under process. And next one is Coda tympani syndrome, later on will come to know what it is. And next, paralytic secretion of the saliva, one side, from the one side there is no secretion, that in the sense the parotid gland, from the parotid glands there is no secretion from the one side. And next one is augmentation secretion of the saliva. And next is mumps. Okay, and next Sjogren syndrome. This Sjogren syndrome is a autoimmune disorder. Okay, and next is come to the stomach. What is this stomach? Okay, stomach is a part which is directly involved in the process of the digestion. Okay, so through the pharynx and the esophagus, this food material enters into the stomach. Okay, so the material which is coming from the mouth is in the form of the bolus. Okay, that bolus forms are like ball-like structures, are enters into the stomach, and then there is a digestion process is starts. Before going to that, what is the functional anatomy of the stomach, we're going to learn. Okay, right. And the volume of the empty stomach is about 50 ml, 50 milliliters. Okay, and it can expand, accommodate to till 1 to 1.5 liters. Okay, and next one is it is capable to expanding still further to the 4 liters. Okay, generally it is about 50 ml and it can expand up to 1 to 1.5 liters, and next one is the maximum expansion is about 4 liters. Okay, and next one is if you see the parts of the stomach, the cardiac region or the cardiac notch it is. Okay, and next one is fundus and the body or the corpus, and next one is pyloric region. The uppermost part of the stomach is called as fundus, and in between that fundus and the esophageal sphincters, end of the esophagus, there is wheel-shaped like structure is present, that is called as cardiac notch. There is an impression of the heart. Okay, and next one is the middle part is called as the body or the corpus it is called as, and next the end part or the last part of the stomach is called as the pyloric region or the pylorus it is. Okay, and this stomach is continued as the intestine part, that is the small intestine. Okay, and next one is if you see the glands of the stomach, there are first one is fundic glands, next one is pyloric glands, and next one is cardiac glands. Okay, 3 types of the glands are present in this. Okay, the pyloric glands are the important glands here. Okay, the fundic glands, pyloric glands, and the cardiac glands. 3 types of the glands are present. Okay, if you see this structure in this, okay, there are different types of the cells are there. Majorly the gastric pit, and next mucus cells, parietal cells, and the chief cells, G cells. 4 types of the cells are present here, which secretes different types of the secretion and the enzymes and the hormones from the stomach. All these are present in the walls of the stomach it is. Okay, and next one is the chief cells secretes pepsinogen, renin, lipase, gelatinase, and the urease. Okay, from the chief cells there is secretion of the pepsinogen, renin, lipase, gelatinase, and the urease. Okay, and next one is the parietal cell secretes hydrochloric acid or the HCl. Okay, so it is a concentrated hydrochloric acid is secreted from the parietal cells. Okay, and next one is mucus cells. Through this mucus cells, the mucin is produced, which is a protective layer of the stomach. Okay, from the hydrochloric acid. Okay, that is the mucin is the mucus layer protects from the hydrochloric acid. Okay, and next is G cells produces gastrin, it is a hormone it is, and next one is enterochromaffin cells, it secretes serotonin. Okay, and next is enterochromaffin cells and like cells that secretes histamine. Enterochromaffin cells, serotonin, like cells, histamine. Okay, and next one is if you see the functions of the stomach. Okay, so here the mechanical functions are 2 types. One is storage function and next one is formation of the chyme. So from the mouth to stomach, bolus, and from the stomach to intestine, that is transported as the chyme it is. Okay, and that is mixed with the gastric juice, and next that is transported as a chyme through the pyloric end of the stomach and enters into the intestine, small intestine it is. Okay, and next one is digestive function, the digestion, how it is going on, so we'll come to know later on. Okay, and next protective function, hemopoietic function, excretory function. Okay, those are the functions of the stomach. And next, if you see the properties of the gastric juice is produced by the stomach, and the volume is about 1200 ml per day to 1500 ml. Okay, 12 to 1500 ml per day, the secretion of the gastric juice. Okay, and next one is reaction it is highly acidic with 0.9 to 1.2, that is because of the hydrochloric acid presence in the gastric juice. Okay, and next one is specific gravity is about 1.002 to 1.004. Okay, and next composition of the gastric juice. If you see the composition of this gastric juice, 99.5% of the water is present and the 0.5% of the solids are present. In these solids, there are two types of the substances are there. One is organic substances and another one is inorganic substances. Inorganic substances in the sense minerals like anions and cations like sodium, potassium, chloride, hydrochloric acid, bicarbonates and the chlorides, phosphates and the sulfates. Okay, and the organic substances are in two types. One is enzyme and other organic substances. The other organic substances are like mucus and the intrinsic factor. Okay, and the enzymes like pepsin, renin, gastric lipase and gelatinase, urease. These are the enzymes present in the gastric juice. Okay, and next one is, if you see the actions, the pepsin is an enzyme, so the activator of this pepsin is the hydrochloric acid and the substrate is the proteins. The proteins are converted into the proteoses and peptones and the polypeptides by this enzyme. Pepsin converts the proteins into proteases and the peptones and the polypeptides. It is activated by the hydrochloric acid. Okay, and next one is gastric lipase. Gastric lipase is acid medium and activated by the acid medium, and next the triglycerides of the butter is converted into the fatty acids and the glycerols. Okay, and next one is gastric amylase. The gastric amylase activated by the acid medium, same as like hydrochloric acid, and starch is converted as the dextrin and the maltose. Okay, and next is gelatinase, and in the acid medium that is activated by the hydrochloric acid, and by that hydrochloric acid the gelatin and the collagen of the meat is converted as the peptides. Okay, and next one is urease. In the acid medium by the activation by the hydrochloric acid, and the urea is converted into the ammonia. And next is hydrochloric acid secretion. The hydrochloric acid is secreted from the parietal cells of the stomach. Okay, right. This hydrochloric acid, how it's secreted in the sense, the water which is present in the stomach and the carbon dioxide after the metabolic phase in the stomach cells, okay, these two combined and it forms H2CO3 in the presence of the carbonic anhydrase. Hydrochloric acid is produced from the parietal cells, and before that how it's secreted in the sense, the water content which is present in the stomach and that is combined with the carbon dioxide after the metabolic waste, the gas metabolic waste, so which is carbon dioxide, and these two combine and in the presence of the carbonic anhydrase, okay, so it makes H2CO3 carbonic acid. Okay, and this carbonic acid is again divided into HCO3 minus and the H in the cell. Okay, and this H plus ion enters into the lumen of the cell, and the HCO3 minus so goes to the into the out of the cell. Okay, right, and this H plus ion enters into the lumen. Okay, and next is sodium chloride which is present in the stream. Okay, that sodium chloride divides into sodium ion and the chloride ion. This chloride ion directly enters into the lumen of the parietal cell, and the H plus ion is present already there. Okay, right, this H plus ion and that chloride ion which is comes enters into the lumen of the parietal cell, and after the dividing of the sodium chloride as the sodium ion and the chlorine ion. Okay, so and this H plus ion and the chloride ion combines and it makes HCl, a concentrated acid. Okay, and that sodium which is divided from the chloride, okay, that sodium and that HCO3 minus, okay, these two combined and it forms NaHCO3, sodium bicarbonate. Okay, right, this is what the secretion of the hydrochloric acid, right. And next one is how this method of study of the secretion of the gastric juice. For this, there is a experimental evidence is there, that is what the Pavlov's pouch, so which is very, very important here. Okay, so this Pavlov's pouch is nothing but they the Pavlov is a scientist, okay, so he made a cut in the body of the stomach of the dog, and he made a pouch like structure, so and that is sutured to that cutted place of the stomach, and that is sutured, and after 3 to 4 days that is innervated, that pouch is innervated, and then so he gives some food to that dog, and that too by the sham feeding process. Sham feeding is nothing but false feeding it is, okay. So that in the sense, so a pipe like structure comes out of the pharynx, okay. Whatever the dog taking food, and that is comes directly out of the through the pharynx, the tube like structure, okay, so that comes outside, but dog has satisfaction of the taking of the food, okay. And at the same time he observed that Pavlov observed the secretions in the stomach, that to this Pavlov's pouch. That is what the Pavlov's pouch it is. Okay. And next one is the somewhat modification pouch, like hidden pouch, and next Bickle pouch, and next feral pouch, and IV pouch, and next one is sham feeding. That's for the all these pouches are this depending upon the sham feeding only. That is what the false feeding. The dog has the satisfaction of the taking of the food, but that is not enters into the esophagus, that comes directly out of the pharynx. Okay, that is what that. And next one is the regulation of the gastric juice. The regulation of the gastric juice in the 3 phases. One is the cephalic phase, next one is gastric phase, and next one is intestinal phase. That in the sense, when the food is in the mouth, that is the cephalic phase, and next one is when the food enters into the stomach, that is the gastric phase, and when the food enters into the intestine, that is the intestinal phase. Okay, right. And first is the cephalic phase. Cephalic phase is in 2 ways. One is conditional reflex and another one is unconditional reflex. Conditional reflex in the sense, when we'll see the food and we'll get the thought of the food, that in the sense vision, thought, smell, and hearing about the food. All these are the conditional reflexes. And at that time, due to this conditional reflex, when we are seeing the food, when we are observing the food, when we'll get the thought of the food, when we'll hear about that particular food, and when we'll get the smell of that food, all these are the conditional reflexes. At that time, there is stimulation of the vagus nerve, and then there is a secretion in the stomach is observed. That is what the gastric juice secretion is started. That is by the conditional reflex in the cephalic phase. And next one is unconditional reflex. Unconditional reflex in the is nothing but when the food enters into the mouth, immediately there is a stimulation of the vagus nerve, and there is a secretion of the gastric juice in the stomach. Okay, the cephalic phase divided into 2 phases. So one is by the conditional reflex, unconditional reflexes. Whatever by the stimulation of the vagus nerve, there is secretion of the gastric juice in the stomach is observed. Okay, right. And next one is gastric juice, when the bolus enters into the stomach. The first phase is completely neural action, that is by the vagus nerve, okay. And the second phase is the both, that in the sense humoral and at the same time neural action. That in the sense, by the action of the gastrin is the humoral action, and by the action of the vagus nerve, that is neural action. So that it is by the neural and at the same time humoral action. Okay, when that bolus enters into the stomach, immediately there is activation of the gastric juice and the activation of the vagus nerve. By the stimulus of this, there is the gastric juice secretion is present in the stomach. Okay, this is what the second one. And when the food enters into the intestine, then the third phase is started. That is what the intestinal phase. Okay, so when the food enters into the intestine, there is no need of the secretion actually in the stomach. isn't it? Okay, so that the gastric juice secretion is inhibited by the two types of the hormones. One is secretin and another one is CCK. That is what the intestinal phase. Okay, so that in the sense what, from the stomach to intestine the food is transported as the chyme, and that chyme enters into the intestine, then the intestinal phase is started, and at that time, so secretin and CCK, cholecystokinin, two are the hormones, and by the action of these hormones the gastric secretion is stopped. Okay, it is completely inhibitory action of the gastric juice, and it is completely humoral action. The phase 1 cephalic phase is completely neural action. Phase 2 gastric juice is both humoral and at the same time neural action, and next one is third phase intestinal phase is completely humoral action. It is by the hormones. Okay, and next one is applied aspects. Okay, the applied aspects of the stomach is gastritis, gastric atrophy, and next peptic ulcer, and after that Zollinger-Ellison syndrome. Four syndromes are the applied aspects of this. Okay, and next one is pancreas. Pancreas is an organ present just below the stomach, and it is also secretes, so like gastric juice type of this secretion, and this juice is called as pancreatic juice. Okay, the pancreatic juice is if you see the properties of this pancreatic juice, the volume of the pancreatic juice is about 500 to 800 ml per day. It is highly alkaline. Okay, and the reaction is highly alkaline. The pH is about 8 to 8.3. Okay, and next the specific gravity or the density is about 1.10 to 1.18. Okay, and next one is, if you see the reactions and the everything of this, so the composition of the pancreatic juice, the water is about 99.5% and the solids are about 0.5%. Okay, these solids are divided into organic components and the inorganic components. Inorganic components are the minerals like anions and the cations like sodium, calcium, potassium, magnesium, phosphorus, sulfates, chlorides, and all these are present, and bicarbonates is also present here. Okay, and next one is in the organic components there are two types. One is enzymes and other organic components. Other organic components are like albumin and the globulins. The enzymes are divided into two types. One is proteolytic enzymes and another one is lipolytic enzymes, and another one is amylolytic enzymes. Okay, the proteolytic enzymes are trypsin, chymotrypsin, carboxy peptidase, and after that nuclease, elastase, and collagenase are the proteolytic enzymes. And next one is lipolytic enzymes are the pancreatic lipase, and next cholesterol esterase, and next one is phospholipase A, phospholipase B, and next colipase, and next is bile salt activated lipase. These are the lipolytic enzymes. And next, applied aspects of this pancreasis, one is pancreatitis and another one is steatorrhea. The bulky and the foully smelled fecus is excreted. Okay, that is called as steatorrhea. Okay, and next, liver and gallbladder. Before going to this, the pancreatic juice regulation is also same as like the gastric juice regulation itself. Like the three phases are present: cephalic phase, gastric phase, and the intestinal phase. Okay, right. And next is the liver and the gallbladder. And this liver is the largest organ, and the hepatic lobes, if you see the functional anatomy of this, the hepatic lobes are present. These hepatic lobes are divided into hepatic lobules, and next that hepatic lobules by the hepatocytes have the hepatic plates, and next portal tracts are present in the functional anatomy of the liver. If you see this, okay, two lobes are present in this, and the gallbladder is also present. Okay, and here the liver is divided into two lobes, right lobe and the left lobe, and the orifices. Okay, and if you see the structure of this, and the hepatocyte is the structural unit of the liver, and it secretes a secretion is called as bile. Okay, and if you see this here, we'll get clear image of the hepatocytes, and hepatocytes are hepatic lobules, and hepatic lobules has hepato lobes, and by that hepatic circulation starts, and the gallbladder secretion, and the biliary system, and next connected to the pancreas, and next that is called as common bile duct, and next that pours into the intestine, small intestine, at the part of the duodenum, in the second part of the duodenum, that is called as ample of water, and the secretion like bile and this pancreas are secreted. It is whatever secreted from the pancreas and the liver, those are poured into the ample of water part of the small intestine. Okay, so and then that mixed with the chyme, and later on we'll discuss about all this. Okay, and come to the biliary system. From that, common bile duct, okay, cystic duct and the common bile duct, and the cystic duct binds with the gallbladder and it makes common bile duct, and next that common bile duct comes out of the liver, and the secretions from the pancreas and the secretion from the liver are both are combined, and next these are poured into the small intestine, second part of the duodenum, and that part is called as ample of water. Okay, this is what the biliary system. And next, enterohepatic circulation. After completion of the chyme process completely, so after the absorption and the bile recirculated to the liver, that is called as enterohepatic circulation. Entero in the sense, enteric in the sense intestine, and hepatic in the sense liver. Okay, so what intestine to liver, again it going back, that's why it is called as enterohepatic circulation. Okay, and next one is, if you see the properties of the bile, the volume of the bile is about 800 to 1200 ml per day, and the reaction is, it is completely alkaline, and the pH is about 8 to 8.6. Okay, and next one is the specific gravity is about 1.010 to 1.011. Okay, and next the color is golden yellow or the green color is present, the bile is in that color. And next one is, if you see the composition of the bile, okay, the water is about 97.6%, and the solids are about 2.4%. Okay, the solids are again divided into inorganic components and the organic components, and the inorganic components are sodium, calcium, potassium chlorides, bicarbonates. Okay, and the organic substances like bile salts, bile pigments, cholesterol, fatty acid, lecithin, and the mucin are the organic substances are present in the bile. Okay, and next one is, if you see the circulation and the what happens and the how these bile salts and the bile pigments are produced, in the sense, when there is the destruction of the RBC, hemoglobin comes out of the RBCs, and that hemoglobin destructed as the heme and globulin. Globulin is a protein part which is goes for the undergo for the protein metabolism, and the heme is a color compound, and this heme part is produces the biliverdin, and next that biliverdin is converted as the bilirubin and enters into the bloodstream. Okay, and after that, this when it enters into the blood, bilirubin enters into the blood, that is called as free bilirubin, and this free bilirubin, if directly comes to the liver, and that is called as conjugated bilirubin. Okay, this conjugated bilirubin through the hepatic circulation via the bile enters into the intestine, and again goes to the, so through the enterohepatic circulation, if it goes back, okay, so that is conjugated bilirubin. And if that bilirubin passed out through the urine, that is called as urobilinogen, and if it is passed through the fecal matter, that is called as stercobilinogen. Through, if it is passed out through the urine, that is called as urobilinogen, and if it is passed out through the fecal matter, that is called as stercobilinogen. Okay, so this is what this process. And next one is functions of the bile. If you see the functions of the bile functions are like digestive function, and next one is absorptive function, and excretory function, laxative function, antiseptic function, and choleritic function, and next maintenance of the pH, and prevention of the gallstones formation in the gallbladder, and next lubrication function, and next one is coagulation function, and next if you see the functions of the liver, it includes the bile functions, and see the functions, and it is the largest gland we know. Okay, it's the metabolic functions, storage functions, synthetic functions, and secretion of the bile, excretory function, and the heat production, hemopoietic function, and heat production, and hemolytic function, inactivation of the hormones and the drugs. Okay, and next one is defensive and the detoxification functions. Okay, and next if you see the functions of the gallbladder, so storage of the bile and the concentration of the bile is increased in this, and that in the sense concentrated bile is stored in the gallbladder. And next one is alteration of the pH of the bile. Okay, so that is the one of the important function of the gallbladder. And next secretion of the mucin, and next maintenance of the pressure in the biliary system. All these are the functions of the gallbladder. And if you see the applied aspect of this is the jaundice or icterus. Okay, so jaundice is nothing but the bile concentration or the bile secretion is more, then that disorder is called as jaundice. This jaundice is in three types. So one is pre-hepatic jaundice, and next one is hepatic jaundice, another one is post-hepatic jaundice. Pre-hepatic jaundice is also called as hemolytic jaundice, and the post-hepatic jaundice is also called as obstructive jaundice. Hepatic jaundice is hepatic jaundice. Okay, so hemolytic jaundice is by the destruction of the RBCs, the hemoglobin comes outside, and that destructed more, and by that the bile concentration is increased. Okay, and by the obstructive, so if there is any injuries or the internal bleeding occurs, then there is a destruction of the hemoglobin, that is by the obstructions. Okay, so that is called as obstructive jaundice. And next one is hepatic jaundice. In case of liver, okay, the bile secretion is more, that is called as hepatic jaundice. Okay, so it leads to hepatitis and the gallstones. The gallstones are nothing but what? So the gallbladder concentration is increased by that the bile forms stone-like structures, so that is called as gallstones. And the hepatitis, these are in four types. Okay, hepatitis A, hepatitis B, hepatitis C, and hepatitis D. Four types of the hepatitis. And next one is small intestine. Another organ in the digestive system is small intestine. If you see this small intestine, so if the functional anatomy, there are three parts are present. The proximal part of the small intestine is called as duodenum, and next one is middle part of the small intestine is called as jejunum, and the final or the distal part of the small intestine is called as ileum. And next, so if you see these structures, duodenum, jejunum, and the ileum, so three structures are present in the small intestine. Okay, and next one is the cells, the microvilli are present in the structure of the small intestine. By that, the succus entericus is a secretion of the small intestine, is secreted by the villi. By the movement of the villi, there is a secretion of the small intestine juice, that is called as succus entericus. Okay, and next one, if you see the properties of this succus entericus, the volume of the succus entericus is about 1800 ml per day, and the reaction is completely alkaline, and the pH is about 8.3. Okay, and next one is, if you see the composition of this succus entericus, and the water is about 99.5% and the solids are about 0.5%. Water is water, there is no dividation, and the solids are again divided into two types. One is organic components, another one is inorganic components. If you see the inorganic components, minerals like anions and the cations are present, like sodium, potassium, calcium, bicarbonates, chlorides, phosphates and the sulfates are present. And next one is organic components is enzymes and the other organic components. The enzymes are divided into three types. So one is proteolytic enzymes, and next one is aminolytic enzymes, and next one is lipolytic enzymes. Okay, the proteolytic enzymes we have peptidase, peptidases, all the groups are called as peptidases, and in that aminopeptidase, and next is dipeptidase, and next one is tripeptidase. Okay, amino, di and tripeptidases. Okay, and next one is aminolytic enzymes are like sucrase, maltase, galactase, lactase, dextrinase and the tehranase. Okay, so and next lipolytic enzyme is the lipase. Okay, and the enterokinase is also one of the component. Okay, this is what the composition of the succus entericus, and other organic substances are like mucin or the mucus. Okay, and next intrinsic factor and defensins, okay, are the present. And next, if you see the functions of the small intestine, it includes also the functions of the succus entericus. In that we have mechanical function, secretory function, hormonal function, digestive function, activator function, and next hemopoietic function, and hydrolytic function, absorptive function. Majorly in the small intestine, that what acidic chyme is neutralized by this bile and the pancreatic juice secretion in the ampoule of water. That chyme is mixed with this alkaline and the food material is neutralized. Okay, so that is what the major function is in the small intestine. And next one is applied aspects is malabsorption, and that in the sense, by the malabsorption the nutritive substances are not absorbed properly. That is what the malabsorption. And next, malabsorption syndrome, and next is Crohn's disease of enteritis, and next tropical spore, and after that steatorrhea, bulky and foully smelled fecal matter excreted, and next is celiac disease. And after that, large intestine. This large intestine is the final organ in the digestive system, and if you see the structure of this, it is continued with the small intestine. The junction between the small intestine and the large intestine is called as ileocecal fossa. Okay, so cecum is the part of the large intestine, and the ileum is a part of small intestine. That is the last part of the small intestine is the ileum, and here the first part of the large intestine is the cecum. So that the junction is called as ileocecal fossa. Okay, and if you see the parts of the large intestine, it is like this type of structure we have. So that in the sense, the part which is essence, that is called as, this large intestine is also called as colon. Okay, the ascending part of the large intestine is called as ascending colon, and the transverse part of the large intestine is called as transverse colon, and which is descends again, that is called as descending colon. So, and which is comes like this type of term, that is called as sigmoid colon, and it is continued with the anal canal and the rectum. Okay, so ascending colon, transverse colon, descending colon, sigmoid colon, anal canal and the rectum. These are the parts of the large intestine. And next, if you see the structures in this, the secretion of the large intestine is called as large intestinal juice. It contains 0.5% of the solids and the 99.5% of the water. Okay, the solids are again divided into organic components and the inorganic components. Inorganic components are the minerals, and the organic components are the albumin, globulin, mucin, urea and the debris of the epithelial cells. Okay, and next one is, if you see the large intestinal functions, important one. Okay, absorptive function, formation of the fecus. okay, and the excretory function, secretory function, and synthetic function. Okay, majorly it absorbs from the waste material, so which is already digested, so it absorbs from that, reabsorption it is, and next formation of fecus. Out after the all digestion process, it excretes the fecus. The digestive waste material is in the form of the fecus, it excretes. Okay, and next secretory function, it secretes large intestinal juice, and synthetic function, it synthesizes the different types of the factors. Okay, and next if you see the applied aspects of this, one is diarrhea, and next one is constipation. That in the sense, constipation in the sense, there is no proper movement of the fecal matter is called as constipation. And next, appendicitis. Appendix is a part so which is useless in case of the human being. So the animals which eating grass, in those it is very active. In case of the human beings, it is a useless part. Okay, so when we are taking the food and while passing through the ileocecal fossa, by the pressure, somewhat food enters into the appendix, and the walls are present on that appendix, and it should not move upwards. Only there is a gate-like structure, so it enters into the appendix. Okay, so but it closes when the food should come outside. Okay, that in the sense the food should not come out of that. Okay, so by that what happens, it completely makes as the poisonous substances, and after that, so there is enlargement of the appendix, and finally so it creates pressure on the side structures, and by that there is medical emergency. If within 24 hours that appendix should be cut. This process is called as appendicitis. Okay, and so that in the sense appendix is appendectomy. By the appendicitis there is appendectomy, in the sense appendix is cut. Okay, so and next is ulcerative colitis, and next one is the movements of the GIT, or movements of the gastrointestinal tract. Okay, in this movements of the gastrointestinal tract, there are totally 11 movements are present. In that, the first one is mastication, in the sense the breaking down of the food material in the mouth into the small particles is called as mastication. Okay, this is by the teeth and at the same time by the tongue. Okay, and next one is deglutition process. Deglutition is nothing but it is a swallowing. It is in the three phases. One is the oral phase, and next one is pharyngeal phase, and next is the esophageal phase. When the food enters into the mouth after the mastication process, the food from the mouth to pharynx, transportation of the food as in the form of the bolus from the mouth to pharynx is called as the oral phase. And next one is the pharyngeal phase. The movement of that bolus food material from the pharynx to esophagus is called as pharyngeal phase. And next phase is pharynx to esophagus is the pharyngeal phase, and after that the esophageal phase, so in which the food material or the bolus, the movement of this bolus from the esophagus to stomach is called as esophageal phase. Okay, and if you see this diagram, so we'll come to know three phases of that. The first one is the oral phase, and next one is the pharyngeal phase, and next one is the esophageal phase. Okay, and next one is the movements of the stomach. Another movement is the movement of the stomach. In this hunger contractions, there are three types of the hunger contractions are there during the after completion of the food processing or the digestion process. There are three types of the contraction occurs. One is contractions 1, hunger contractions type 1, hunger contractions type 2, hunger contractions type 3. Okay, and next filling of the stomach. So after completion of this process, when we are taking the food at the pyloric end, that in the sense in the stomach we have three parts: fundus, body, and the pylorus. At the pyloric end there is a sphincter is present that is closed, and the upper part is also one sphincter is present that is open. Esophageal sphincter is open, and the pyloric part, that in the connection between the small intestine and the stomach is sphincter is present that is closed, and then the filling of the stomach is present. Okay, and after that, emptying of the stomach. In the emptying of the stomach, the upper part, the esophageal sphincter is closed, and the pyloric end, the sphincter is opened, and the food material is transported into the intestine, small intestine, the duodenal part. Okay, and next one is vomiting. That in the sense, the movement of the food material upwards. Okay, this is called as reverse peristatic movements. By the reverse peristatic movement, this vomiting is possible, like in the sense it is a reflex process. Vomiting is okay. This vomiting is by the when the food material comes out from the upwards, moves upwards. Okay, so by the mass peristatic movements. Okay, so then there is the food material comes upwards, and then through the mouth it comes outside. This process is called as vomiting. Okay, and next one is movements of the small intestine. Is the 4 types. These movements of the small intestine, first one is mixing movements, and next one is propulsive movements, and third one is peristatic movements in the fasting condition. This is also called as MMC, micromotor complex conditions. Okay, so and next one is movements of the villi. Okay, the mixing movements is in 2 types. One is segmental movements, and another one is pendular movements. By this, the food is mixed with the alkaline medium like pancreatic secretion and the liver secretion. If the chyme is mixed with these 2 secretions, that is called as mixing movements. And next one is propulsive movements. The food is moved forwardly by the propulsive movements. One is peristatic movement, and another one is peristatic rush. Okay, and next one is peristatic in fasting conditions. Okay, so it is also called as migrating motor complex movements. It is okay. So when the stomach is empty and the intestine is empty, then the whatever is the food material present in the large intestine, that comes into the small intestine, and then the movement of the small intestine and the reabsorption of the food, and then it passed out again into the large intestine. This type of movement is called as peristatic movements in the fasting conditions. Okay, and next one is movements of villi. The movements of villi, so by this there is a succus centricus secretion. Okay, so by the movements of villi there is a succus centricus secretion. And next one is movements of the large intestine. In this, the mixing movements by the segmentation contractions, and the propulsive movements by the mass peristatic movements. By this mass peristatic movement, the fecal matter is excreted outside, and the defecation. Defecation is nothing but the excretion of the fecal matter is called as defecation. Okay, and next one is evacuation of the gases and from the gastrointestinal tract. It is in 2 types. One is belching, and another one is flatulence. Belching is through the mouth, the gas comes outside, and the flatulence is the one in which the gases comes outside by the anal canal and the rectum. And if you see the hormonal reactions, okay, so one is gastrin. So the action of the gastrin is it stimulates the gastric juice secretion, and next one is secretin. It stimulates the intestinal juice secretion and it inhibits the gastric juice secretions. And next one is cholecystokinin. It inhibits gastric juice secretion, and at the same time it stimulates the intestinal juice secretion. And next, gastric inhibitory peptide, which inhibits gastric juice secretion. And next one is vasoactive intestinal polypeptide, which involves in the process of the digestion in the small intestine. And next is glucagon, which increases the blood glucose levels. And next one is lecithin. It increases the blood sugar levels. And next one is glucagon-like polypeptide. It is 1-GPL glucagon-like polypeptide it is. And in this, there is the stimulation of the insulin secretion and the inhibition of the gastric motility. from the pancreas. Okay, and next one is glucagon like polypeptide 2. So there is suppression of the appetite. Okay, and after that somatostatin, so which inhibits the and the secretion of the growth hormone, and at the same time there is a balance in between the insulin and at the same time glucagon, it maintains somatostatin. And next pancreatic polypeptide, which increases the secretion of the glucagon and decreases the pancreatic secretions. Okay, and next one is peptide YY. So it is from the L cells of the ileum and the colon, and it inhibits gastric secretions, and the motility is inhibited of the intestinal secretions. Okay, and next one is neuropeptide Y. So it increases the blood flow in the enteric blood vessels. And next one is motilin. So in this there is accelerate the gastric emptying process. Okay, and after that substance P. This substance P is increases the movements of the small intestine. And next one is ghrelin. So by this ghrelin, it promotes growth hormone releasing induced appetite, and the food intake stimulates by the gastric emptying. And next one is carbohydrates are digestion of the carbohydrates, proteins, lipids. Okay, so in the mouth, the saliva, so in that the substrate is polysaccharides, and the starches are digested, and the end product is disaccharides and the dextrin and the maltose. Okay, and next one in the stomach, gastric juice. So by the gastric amylase is the enzyme, and the polysaccharides are converted into the disaccharides, and the sucrose sucrase is an enzyme which converts the sucrose into the glucose and the fructose. And the next one is maltose and is converted into the glucose. And next one is lactase, which converts the lactose into the glucose and the galactose. And the next one dextrinase, which converts the dextrins and the maltoses and the maltorises into the glucose in the form of glucose. And next is trehalase. So in this trehalase, is the end product is the glucose. And next one is the proteins in the mouth. The juice is saliva, and there is no proteolytic enzymes. And in the stomach, gastric juice, pepsin, the proteins are converted into the proteases and the peptones, large polypeptides. And in the small intestine, by the pancreatic juice, trypsin converts the proteases into the dipeptase, tripeptase, and the polypeptases. Okay, and next one is succus entericus. So it is a secretion of the small intestine. In this there is dipeptidases, tripeptidases, amino acids, aminopeptidases. All these converts into the converts dipeptidase, tripeptidase, large polypeptides, converts into the amino acid form. Okay, and this is what this. And another one is lipids. The triglycerides, all are triglycerides and the cholesterol, and all these from the mouth, stomach, and in the small intestine by the secretions of the saliva, gastric juice, pancreatic juice, and the succus entericus. And the end products are fatty acids and the glycerols and the phosphorylcholine free fatty acids. That is what the lipids, and this is what about the completely digestive system as per my knowledge. Thank you very much.